Hi scholars, I'm Okoifa Farida, your personal online study buddy, and this is Share Your Jotter segment. So this is the segment where listeners of the podcast would send me a picture of their jotters on a particular topic in zoology. Now what I would do with these jotters is that first, they will be posted on our social media platforms, and it will be useful to both current and incoming students. Now see, imagine being such a legend that even your jotter is saving lives. Like it's actually literally saving lives. Now the second thing I would do with your jotters is that I would extract the important details out of each of them and compile them into a single jotter, which I like to call the master jotter. This master jotter is what I'll be reading to you today. Therefore, this segment involves extracts of everything worthy of noting on a particular topic from various amazing people with their different shapes and sizes of jotters and handwritings. Now, I mean it when I say different shapes and sizes of jotters and handwriting because handwriting applies to me. I have a terrible handwriting. I write like a chicken. Alright, that's enough shaming myself. So if you're on the road walking, doing chores, bored in the house and in the houseboard, going for exercise, about to sleep, in the bus, on your way to write an exam or a test, or your way to class, then this is the segment you should tune into. This is not me reading a note. That would be way too long for comfort. This is me sharing a jotter.
I'm gonna set your place on fire. Let's get higher.
Protozoa. Protozoas are the first animals or first order of organization. They are single-celled or unicellular organisms. Some live in colonies and some are syncytial. And by syncytial, I mean that there is fusion of two or more cells to form what is called a syncytium. They are microscopic in size, that's 3 to 1,000 microns. They have no germ layer, tissue, or organs. However, specialized organelles are present. There have been over 64,000 species that have been described, and only 50,000 out of those are free living. Locomotion is by Pseudopodia, Flagella, or Syria. The symmetry, they have all types of symmetry. Name it bilateral, radial, spherical, or asymmetrical. Now they are free living organisms. It could be commensal, parasitic, or mutualistic. They are mostly naked, but few have simple protective exoskeletons called tests. Examples of these organisms or protozoas that fall in this category are Arcella and Diphagia. Now, nutrition could be autotrophic, saprophytic, saprozoic, or holozoic. Now, the specific type of autotrophic nutrition that they take part in is called holophytic nutrition. So they take part in holophytic autotrophic nutrition. Reproduction, it could be sexual or asexual. Let's go first to asexual. Asexual reproduction in protozoa are divided into two. It could be by budding or by fission. Now, I'm explaining fission. Now, we all know budding, how to grow a bud, external part of an organism. But fission is the division of cells. By fission, now, there's binary fission and there's multiple fission. Now, binary fission is division into two identical cells. We all know that. Under binary fission, the kind of asexual reproduction they take part in is longitudinal and transverse binary fission. While under multiple fission, where it is divided into multiple parts, there are different there are also two types, two subcategories, and they are schizogony and spirogony. So basically, types of multiple fission, schizogony and spirogony. Schizogony is the multiple and rapid division of cells into merozoites, while spirogony is spore formation, where multiple sporozoites are formed. Now you might be asked, because I remember there was an assignment where we were asked to list the difference between schizogony and spirogony. What I got is definition, but like another definition. So if you didn't get the first definition, this is another definition for you. Spirogony is a spore forming stage of the protozoan life cycle that produces sporozoites outside the host. Now note that outside the host. While schizogony is an asexual multiplication stage of the protozoan life cycle, which allows multiplication of the sporozoites inside the host. So if you notice, one is outside, one is inside. One that is outside the host is spirogony. One that is inside the host is schizogony. That could help you remember. Now, moving to the sexual reproduction of protozoas, we have sexual reproduction of protozoas. take part in conjugation, which is the exchange of gametic nuclei between two cells. Example can be found in Paramecium sp. Orthogamy, which is the fusion of two gametic nuclei within the same cell. And syngamy, fusion of two gametes form two different protozoans. So let's say it again. Conjugation, exchange of gametic nuclei between two cells. Example, Paramecium sp. Orthogamy, fusion of two gametic nuclei within the same cell, while syngamy, fusion of two gametes from two different protozoans. Okay, now classification of protozoans, we have phylum Sarcodina, which is examples are amoeba, entamoeba, arcella. We have phylum Mastigophora, examples are Euglena, Chlamydomonas, Trypanosoma, Trichomonas, and Trichonympha. And the third one, phylum Epicomplexa, examples are Plasmodium, Elimera, Toxoplasma. And the fourth one is, and which is the last, phylum Celiophora. Examples are Paramecium and Balantidium. Try to list them again in your head. I'll give you a short pause before I start taking them one by one.
Hello! Since you guys refused to greet me, I decided to come and greet you. To those of you that don't know who I am Jack. And I'm basically your tour guide, stroke, study buddy for this podcast. I'm literally just here for you to make sure that you are paying attention and enjoying it at all times. You'd hear my voice often. I hope you like it. If you don't like my voice, get used to it already. Because you're going to hear me every time. In case you're dozing off, it's me that will tell you to wake up, scholar. And it's me that will be in charge of making sure that you're at alert at all times. And just get used to having me around. Yeah, once again, I'm Jack. When you hear, oh yeah, come, let's Jack, basically, you're talking about me. So, come, let's Jack. Quick. Alright, guys, it's supposed to be our break time, our happy hour segment, where we have other or any type of features, where we feature any interesting thing. But for now, today, this episode, my boss Farida, she said she's the one that wants to launch it, so I am allowing her to talk to us very briefly, but this will happen again. Trust me, the next time she comes and tells me she wants to join my happy hour segment, I'm going to chase her away. Anyways, she's going to talk to us right now, and let's just have fun and then listen to her. I'm sure she has a lot of interesting and nice things to say.
Alright, so I'm literally recording this without a script or a microphone or anything. I'm just recording it as myself. And I just want to use this particular happy hour to just talk about my own personal feelings. Not necessarily feelings. I actually mean like what I think and just my message to you guys. And basically, this podcast is not really. It's not obviously it's not perfect because it's the first episode. I'm really nervous because before now I've been like I've had the idea of it for a while now, but like I never really did it. But then I summoned the courage to actually just do it, and I appreciate every review and every support and every person that is participating or listening or just appreciating it or learning from it in general. And just know that I really appreciate you, and you guys are the reason I continue doing this. And so basically what I want to say is that just know that this podcast, it might not be the best, fine, it might not be perfect, yes, but there's room for improvement. We're going to improve as the episodes go on. And that's just it, because I'm saying this because to be honest, the reason why I was very reluctant is because I was so scared of not being perfect that I actually forgot that if you don't do it, then you can't actually grow. And actually just decided that, okay, yeah, so let me just do it. Let me rough it, like we always say. And so I'm roughing it. So I hope it's actually beneficial. And if it's not really perfect, you can actually just tell me. You can comment, or if you have any of our contacts or any of our social media pages, you can comment nicely because I'm a human being. So just comment and like ways you can improve it. If you want to partake, there are ways to partake in it. Yeah, so there won't be this kind of, I won't be talking, don't worry. The next happy hour, you won't hear me talking just randomly about my life and telling you the story of my life. You'll find actual interesting things. We have things like random facts with a very wonderful scholar. His name is Proficient Style Bender. We have grammar check with Kembiso. It's going to be like it's your fixogrammar, it's a like sub-segment. And it's like you use that to just learn how to speak better. Just because it's interesting and it's good to just be able to communicate with what you have in mind effectively. Then, and also, so you don't get shamed when you're asking anyone out, like if you're a boy. Next, we also have other things. If you want to feature in the happy hour segment, it's really not hard. You can just all you have to do is send a message via any of our social media platforms. And also, apart from featuring in the segment, if you have maybe something you want to display, maybe you have a business, you could just DM us for ad. And or if you have a project that you're working on, or you have an announcement, or even better, you have a talent that you like to, like probably a poem or spoken words or anything nice and interesting that you like to share, then this is the sub-segment that would fit you into. And so you could just send a DM via any of our social media platforms. We have Instagram and WhatsApp for now. So basically, yeah, what did I even say? I don't remember. But I just know that the main thing I want to tell you is that this is the first episode, so it's not going to be perfect, but we are aiming towards perfection. We are getting there, and I just want everyone to learn from it, because that's the major thing. Learn from it while having fun. I want you guys to listen to my voice when you're walking, when you're doing any like random thing. Like I want that like my voice to be I want to be your study partner. I'm actually literally begging you, allow me to be your study buddy. Yeah, so it's been a pleasure having you listen to me. I hope my voice isn't too either deep or I don't know. I actually love my voice. I'm absolutely in love with my voice. But I don't know if you have a problem with my voice. Just try and reduce the volume or increase the volume depending on the one that fits into your need. Yeah, so thank you very much for listening. Once again, I'm Okwaifa Farida, and I'm forcing myself to be your online study buddy. And I'm telling you, we are come. Let's jack. Thank you very much. Now, phylum Sarcodina, they are called rhizopods. They are amoeba-like organisms. Examples are amoeba, entamoeba, and arcella. I've said that before. They are organisms that move with the aid of the Pseudopodia. They are free-living. They are rarely parasitic. They have single nucleus, monomorphic nuclei. They capture food via food cup using the Pseudopodia. Pellicle is absent. And you know the pellicle is responsible for the shape of organisms. So when the pellicle is absent, they have variable shape. Their asexual reproduction takes place by binary fission. Example can be found in amoeba. And examples of phylum Sarcodina are amoeba, which has no test, entamoeba, which has no test but is free-living, and arcella, which has a calcareous test but is also free-living. Now, we are going to talk about the Pseudopodia. Now, the Pseudopodia are simple extensions of the cell cytoplasm, has a rigid ectoplasm. The Pseudopodia, they are simple extensions of the cell cytoplasms. Now, the cytoplasm in this cell have rigid ectoplasm in a gel state and a less rigid or fluid endoplasm in a sol state. Now, alternation of this sol and gel state gives rise to what we call the pseudopodial extension. Types of pseudopodia are lobopodia, philopodia, reticulopodia, axopodia. Now, the names are they are quite explanatory. Lobopodia, large and blunt. Example, amoeba. They have large and they look the way they look physically. They are large and blunt. Lobopodia. You can take the L and the B. Philopodia. They have thin extensions. Example, amoeba. Reticulopodia. They have thin extensions which join to form a mesh. Example, foraminiferans. Then we have axopodia. They are long, thin, and supported by axial filaments. One more time. Types of pseudopodia are lobopodia, which are large and blunt. Example, amoeba. Philopodia, thin extensions. Example, amoeba. Reticulopodia, they have thin extensions that join to form a mesh. Example, foraminiferans. Now, axopodia, they are long, thin, and supported by axial filaments. Now, we are going to the second subphylum of Protozoa, which is the Mastigophora. They are called flagellates because they move with one or two flagella. They have only one nucleus, hence they have monomorphic nuclei. Pellicle is present in some, which gives them a definite shape. Asexual reproduction by longitudinal binary fission. Example is Euglena, Trypanosoma, and Leishmania. Some are capable of photosynthesis due to the presence of chloroplasts. Others lack chloroplasts, hence are mutualistic or parasitic, and they are mostly bilaterally symmetrical. Types of Mastigophora, we have Phytomastigophora, example is Euglena and Chlamydomonas, and Zoomastigophora, example is Trypanosoma, Giardia, Trichomoniasis, and Trichonympha. Phyto should give you an idea of plant-based, or those that use autotrophic, or just those that have chloroplasts, while Zoo should give you the idea of animals, or just like those that don't have chloroplasts. Now, examples of Mastigophora now. We have Trypanosoma. Trypanosoma causes sleeping sickness, and these organisms, they are only heterotrophic, they don't have chloroplasts, and they are all parasitic. Leishmania sp, it causes leishmaniasis. Giardia causes stomach infection, which is called giardiasis. Volvox sp, they are colonial species. So I mentioned 4 examples of Phytomastigophora. We said Trypanosoma, which is sleeping sickness, Leishmania, Giardia, and Volvox sp. Now moving on, Epicomplexa, phylum Epicomplexa. Phylum Epicomplexa, or Sporozoids. They have no locomotive structures, hence no movement. They have one type of nucleus, hence they have monomorphic nuclei. All species are parasitic. Adult movement is feeble. Feeding organelles are lacking. Now, from all these things, you know that they are parasitic because they don't have feeding organelles, they can't move. So hence they have to depend on others. Now, pellicle is present, hence they have a definite shape. At least they have that one. They have their asexual reproduction involves multiple fission, that's Schizogony and Spirogony. Examples are Plasmodium, Eimera, and Toxoplasma. Let's talk about Plasmodium. We all know Plasmodium causes malaria, but there are types of Plasmodium, and it's important for us to know them. We have Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, and Plasmodium falciparum. Now, Plasmodium falciparum, which is the last one I mentioned, is the most virulent and has caused over 50% of the global malaria cases in the world. Toxoplasma gondii. Toxoplasma gondii is another example of organisms that can be found in Epicomplexa, in subphylum Epicomplexa, and it's also called T. gondii. It causes toxoplasmosis, which is an opportunistic infection in immunocompromised people. An example can be found in HIV and AIDS patients. Now, toxoplasmosis is transmitted by contact with dogs. We are moving to the last phylum, which is phylum Celiophora. They are called the ciliates because they move with the aid of the cilia. They are free-living, rarely parasitic. They capture food with the aid of the cilia. Usually, they have 2 sizes of types of nuclei. That is the micronucleus and the macronucleus. From the name, you know that one is big and one is small. They reproduce asexually by transverse and binary fission. Sexually, they reproduce by conjugation. Example can be found in Paramecium. Examples of ciliates generally are Vorticella and Stentor. More examples are Paramecium and Balantidium. If you don't kind of forget what Balantidium is, remember Balenciaga. Yeah. So now, Flagella and Syria. Now we are going into Flagella and Syria specifically because we mentioned them over and over. In Flagella, they are larger than cilia, but both of them have similar internal structures. How does the internal structure look? Each part of their length contains 9 to 2 arrangements of the microtubules called axoneme, which strengthens it. Now, what I mean by 9 to 2 is that there is 9 pairs of peripheral or external, which is found outside, and 2 central tubules found inside. Now, we are going into the ecological importance of Protozoa. Beneficial, they have the unicellular level of organization, they have highly specialized organelles for various physiological processes. They are the most prominent members of the aquatic food chain, especially detrivores, because detrivores are the primary source of food, because they are like the plants of oceans. That's all. I like to say that jotters are like the man-made version of the book of life. Each jotter tells the story of its owner. Each spill of ink on every page shows the sweat that dropped from the person's hard work. Each mark shows the person's resilience and dedication, and each jotting is a memory from a time in our lives. That's why we revisit our jotters over and over again. It's like we stole that moment and sealed it in a paper. The more you revisit, the more you remember. Each jotter should last forever, and that's why you should share your jotter. Hi scholar, I'm Okpefa Farida, your personal online study buddy, and I'm saying to you today, share your jotter. All the information used in this episode are from my jotter and are sourced from various authentic materials, some of which are Unilab recommended ZLY lab manual for 100 level students, ZLY 101 slides, ZLY notes, Google, cherry picks from benzene tutorial because that's the tutorial I attend. By the way, there's a crash course stroke revision class that is ongoing at benzene tutorial right now. If you think you need aid or assistance for you to push, feel free to find Dr. Beniduli's number and just send a DM to him and just find a way to join the lesson because it's amazing, to be honest, and I'm talking from experience. Yeah, so this episode of Oya Come Let's Jack is especially dedicated to my family. That is my dad, my mom, and my 3 siblings. They have supported me financially, morally, and spiritually. And without them, this podcast won't exist. Thank you very much for listening. Do have an amazing.
Hey, scholar, not so fast. Before you leave, first answer a few questions. Give me examples of organisms under the phylum Sarcodina. What about the phylum Mastodifera? I'm waiting, I'm listening. Could you answer that? If yes, then that's wonderful. Can move on now. If no, then don't go anywhere yet. Go back and replay this podcast so you can learn it and understand it. Okay, so if you enjoyed this episode or you did not enjoy it, feel free to comment on our social media platforms. If you're listening to Anchor, you know that you can actually send a voice note. Yes, you can. Just send a voice note via Anchor and it could even be featured on our next episode. Who knows? I'll see you next episode. Do have a lovely day, guys.
Gonna set your place on fire.